So much for sticking around through the AV difficulties. So my name is Jonas, and I'm a computational biologist at Phage Genomics. We are a biotech company based in Seattle, Washington, that specializes in Hi-C-based technologies related to genome assembly, cytogenetics, and microbiome research. And I work in the microbiome department of the company, where we develop computational methods to analyze and leverage Hi-C metagenomic data. And today I will be talking about a project we're working on where we are developing deep learning models to understand the dynamics of phage-host interactions in metagenomic communities. So first, we heard a little bit about this earlier, but I wanted to give a brief overview of what Hi-C metagenomics is. So one of the fundamental challenges in metagenomics is to deconvolve the diverse mixture of genomes in a metagenomic sample. And Hi-C technology greatly mitigates this because it provides a signal between different contigs in a metagenomic assembly that can be used to group genome fragments together. And this signal is created by chemically cross-linking DNA within a cell, fragmenting the DNA, and then ligating the ends together to create chimeric reads that map to different contigs in a metagenomic assembly. Oh, weird. There we go. One of the most valuable aspects of this technology is its ability to efficiently annotate phage-host interactions in a metagenomic community. Because the library prep cross-links DNA within a cell, Hi-C signal is created between microbial hosts and the mobile elements present inside them. So this signal is able to be used to determine which phage genomes are infecting specific microbial hosts. And this is done for all of the phages and microbes in a sample, thus providing a comprehensive view of interaction dynamics in a metagenomic community. And samples taken from highly diverse communities, such as rumen or wastewater, such as this one is, they can yield upwards of 1,000 phage-host interaction annotations from a single sample, making this process incredibly efficient compared to other techniques. And so a couple of years ago now, we realized just how limited the number of phage-host interactions there are present in the world. So we proposed that using our technology, we should try and collect as many samples as possible from different environments and begin compiling a database of phage-host interactions. So we proposed this idea to the Gates Foundation and the NIH, and we were given funding to do this. We developed partnerships with researchers all over the world, providing them with Hi-C data and analyses to help accelerate their own research, and began building a database of phage-host interactions and annotations. At the time, we estimated that there were roughly less than 5,000 dereplicated phage-host annotations in public databases, and as of now, we have approximately 200,000 interactions, and we intend to continue growing this repository. So the second component of our proposal was to utilize the phage-host interaction data we accumulate to train deep learning models to predict these phage-host interactions independently of that Hi-C sequencing data. And there are a few different ways to format this task, but for the model that I'm talking about today, the task is defined as a binary classification problem. In this approach, the model takes a phage and a host genome as input and predicts the probability that the phage infects the given host. For training, we use our highest quality Hi-C-derived phage-host interaction annotations as the training labels for this dataset. So for the first component of this effort was to develop a way to create a numerical representation of a genome. This essentially requires finding a balance between preserving the valuable sequence context information while also maintaining computational tractability. And so like many in the field of computational biology right now, we turn to the field of natural language processing. And we found that defining the proteins that make up a genome as tokens, such as words are the tokens in a sentence fed to a traditional NLP model, strikes a good balance between information preservation and computational feasibility. And we utilize protein language models to create protein embeddings of these tokens, which then can be fed into the deep learning model. And I also wanted to point out that this embedding technique has been kind of utilized by other individuals, and I most notably want to point out Yunhua Huang, whose work I find highly influential, and I encourage you to go look at it. So for the classification model, I developed a transformer-based architecture. In this step of the network, the phage and host embedding sequences are fed into separate transformer encoder blocks. And these blocks weight the protein embeddings based on their context in the task of predicting the phage-host interactions. The outputs of each encoder are separately mean pooled and then concatenated into separate channels. And I've tried a few variations on this architecture, but I've just generally found that incorporating the separation of the different input data types into the architecture itself tends to help the model training. And I found that if the model is implicitly aware of the boundary between the different input data, it's easier for it to learn information about the relationship between them. So in the final component of the network, the outputs of the transformer encoders are fed into a 2-channel, one-dimensional convolutional neural network. And the CNN scans the phage and host input sequences, extracting additional information about the relationship between the phage and host genomes. The CNN output is finally fed into a fully connected layer that returns the probability of the phage infecting the host. So in summary, we create embedding representations of the genomes using a protein language model. We pass these sequences of embeddings into transformer encoders to create encodings of the input genomes. And we then feed or fuse these encodings into a 1D CNN as separate channels to extract additional information about the relationship, finally passing this output into a fully connected layer to obtain the probability of the phage infecting the host. So for some validation results, this test data set consisted of phage-host interactions taken from high-C metagenomic samples that were not included in the training set. And then additionally, to prevent data leakage, I removed all genomes from the testing set that shared greater than 95% ANI with genomes in the training set. And on this particular data set, we observed an AUC of 0.94 and an average precision of 0.93. And I also wanted to show performance on an orthogonal data set that was not derived from high-C metagenomics. So this data set was taken directly from the publication by AF and includes interactions obtained from public databases. On this data set, we see slightly reduced performance, which I think is likely due to the different Phi annotation methods, but kind of curious about that. So I've talked now about using high-C technology and machine learning for phage-host annotation, and I wanted to quickly touch on the application we are utilizing this technology for. So we're now working on extracting lysine proteins from these phage candidates and testing their antimicrobial potential. Our vision is essentially to be able to utilize this technology for any problematic infections to help combat the growing threat of antibiotic resistance. And thank you so much for taking the time and sticking around to listen about this. If you would like to learn more about our company or hear more about this research, please come talk with me. Thank you.
Thank you.